Okay. Morning, everyone. So what I want to do today is finish off the discussion that we started last class, or I guess lecture, uh, about the carbohydrate handling enzymes, and start a discussion about peptides. And again, I'm really hoping it's going to be the peptide one again. A lot of back and forth, so we're just going to try and really interrogate this because I know we're all familiar with proteins and peptides, and really think about what we mean with those terms and what they do. Okay, so at the end of last class, I left off with this, and I was suggesting, hey, can we think about mechanistically how we can consider an inverting active site would function? So again, this is alpha, UDP. I'm gonna have to go back and check my notes because UDP is glucose UDP is always alpha, and I can't remember if I drew it that way. I really hope I did. So if you ever get a beta glycoside in carbohydrate chemistry, if there's any beta glycoside in nature, needed an inverting enzyme, glycosyltransferase. So the glycosyltransferase is getting its name because it's transferring a glycosyl group, the one in red, onto another molecule. And enzymes always work by adding one monosaccharide to a growing oligosaccharide. So they add them kind of one monosaccharide at a time to the oligosaccharide, and they can target which OH of the oligosaccharide it adds to based on which glycosyltransferase it is. Every glycosyltransferase is specific for a given glycosyl acceptor and a given glycosyl donor. Given. So by definition, if alpha in nature needed a non-inverting glycosyltransferase. Okay. So thinking about this and how this looks, any suggestion on the mechanism that might occur for this inverting reaction? I'm going to draw out the first. I can't because I've saved this image. I'm going to overwrite my image by writing in white and then writing in red. That's going to be a monophosphate then because we've drawn a phosphate already. So what we want to do with the enzyme is we want to turn this UMP into an even better leaving group. It's a pretty good leaving group to begin with, but is there something there that we can use to turn into an even better leaving group? Oh, it sounded like for a second there you're going to bail me out, Hazel.
No, sir. Actually, I was lost for a second. So what did you say?
I was saying, is there, or Ronaz or Nelson, is there anything close to this phosphate that could turn into an even better leaving group than it is? Like phosphate's a good leaving group, but can we make it even better with something that's present on the slide?
With the oxygen?
Well, it's got a lot
Wait.
of oxygens.
Yeah,
So we can form a partial positive charge here because we can make this hydrogen bond to this aspartic acid. So again, what we're seeing, almost all enzymes work through general acid and base catalysis by using specific residues that are well positioned. So we're going to make this oxygen slightly more positive by forming this hydrogen bond with this. Similarly, we can make, I'm going to do the same trick here. We can make this this oxygen slightly more negative because we can form a hydrogen bond with that glutamic acid. So we have got an acid catalyst, we have a base catalyst. And again, in normal situations, normal solution phase chemistry, you can't have acid and base catalysis at the same time because your catalysts will neutralize each other. Enzymes are different. Things are held spatially apart. They can't interact. And so you can have both base and acid catalysis at the same time. That's one of the reasons why enzymes are so efficient. There's no chemical way to do this. So if we're doing that, we're making this slightly more positive and this slightly more negative. It makes sense that we can probably get arrows that look like that. So we're able to make that bond because we're able to activate both the nucleophile and the electrophile with an acid and a base in the enzyme. And we're using the same residues we just used for the glycosidases, but now we're using them to make a bond. Similarly, if we want to talk about the non-inverting, excuse me. So again, we're going to set this guy up the same way. Don't have a choice. The UTP looks like that. And whenever you're doing a non-inverting enzyme, and whenever you're doing any of these enzymes with these glycosyltransferases, they all look the same. They all have the same active sites, the two carboxylic acids. It's the aspartate and the glutamate. Now, we want a non-inverting thing, so we want our sugar to come in alpha, but currently our alpha site's really, really hindered. So it makes sense that, again, we activate the UTP with the oxygen, but now in these enzymes, this glutamate residue is a whole lot closer to this site. And so it acts as the nucleophile to displace the UTP. And so the UTP then floats away. So in this case, we do have a third residue that's involved. There's a histidine residue. It's not far away. I want to make sure I draw the histidine residue right. I'm going to draw it slightly different place here, just to make this really clear how this works. So this, when I just showed there, I showed that glutamate residue was deprotonated. And a reason normally why you get that kind of situation is normally acids are deprotonated by bases. And the standard base to do deprotonation in a lot of biology is histidine. So this hydrogen here initially came from the glutamic acid. So now exit UTP, enter monosac. Whatever monosaccharide it is. We'll do a galactose here. We'll have a C3 galactose. Just because it doesn't matter. It could be any sugar. So this O minus, of course, is close to this hydrogen. And so we can get a hydrogen bond there, which is then going to make this guy particularly nucleophilic. This hydrogen here, this positively charged hydrogen, is close to this carbonyl, which is going to make this whole thing slightly positively charged and a really good leaving group. Acetate's an okay leaving group. Acetic acid is an even better leaving group. And so in this case, if we're drawing all the arrows that we can draw here, we could draw the base deprotonates the hydrogen. The oxygen-hydrogen bond then attacks the carbon-oxygen to do an SN2. And basically all these three residues get involved as well as the sugar. And we get our resulting product, which is now we have our sugar. Our enzyme has now protonated the aspartate. So we've returned this to the original form it was up here. So we'll reset the enzyme. We have reprotonated the glutamate, and we have deprotonated the histidine. Now the histidine, of course, can then go right back and deprotonate that glutamate again and reset the glutamate. So we have these three residues that have been involved in this catalytic system. Sorry, I am allergic to the dust. There's a lot of big work being done upstairs, so give me a second here. So what we've done with these inverting enzymes here, if we notice, this one at this step here, the enzyme is immobilized. Covalently attached. to the sugar. So if we're thinking about this, of these intermediates I've drawn, the starting material, the intermediate, the product, and think about what we talked about the enzymes last class, which of these three components is most tightly fitting to the enzyme? Ignoring the covalent nature of the bond. Maybe this isn't a great example. Maybe I'm going to go back to this example here. At what point in this reaction is that sugar most tightly bound to the enzyme? At what point does it see the greatest benefit of being bound to the enzyme?
When it's an intermediate.
Yeah, it's in the intermediate. It's really the transition state leading to the intermediate. So if I'm going to build an inhibitor for an enzyme, so in the intermediate, this center goes more or less flat. Like in the intermediate, we essentially have a pseudo, we don't quite go through an oxocarbenium, but this goes kind of flat through an SN2-like thing. So if I'm going to build an inhibitor for an enzyme, I want my inhibitor to look like the transition state of the reaction, because transition states bind really, really well to the enzyme. The problem is transition states are unstable. But if I can make something that's stable, that looks like a transition state, the enzyme is going to bind that really well. And the enzyme relies on it being a transition state. The enzyme relies on being transitory. You know, it goes through that state, it either falls back to the starting material or it goes forward to the products. We can really fuck with enzymes by creating things that look like transition states that aren't transition states. So one of the main, so if we think about this and a practical application of this, so influenza works at a whole bunch of, so it's a virus, right? It works by binding to your cells, entering your cells, hijacking the machinery of your cells to replicate, sending out more virions back into your body, which then bind to other cells, hijack the cells, and figure out ways to replicate. How can we inhibit viruses? Like, what are our choices if we're trying to design a drug? So let's say I give you a virus and I say, here's COVID-19. Build drugs against COVID-19. Where are your options for building drugs against a virus? What are your possible targets if you're targeting a virus?
The virus itself.
Okay, so what do you mean the virus itself?
Glycopeptide on the surface of the virus.
So what do you want to do with that?
That would be the chart. Destroy it.
So how do you destroy it?
What's the word? Lysozyme.
Yeah, you can destroy a virus with a lysozyme, but lysozymes are inside cells. So it's very risky to try and bring a virus inside a cell, because that's exactly what the virus wants you to do. So if you want to target, so one option is the virus particle itself. So let's call this like, this is a little bit of a digression, I guess, but it's an interesting digression. The virus envelope. This generally gets targeted with antibodies. And the idea is that if we can generate some, so if you've heard about the Regeneron therapy, that, really, it takes a week for the factory to develop enough antibody to treat 3 people. So it's not really a viable mass-produced kind of thing. And Trump got it when he got the virus, and then said, hey, look, I'm doing great, and that's wonderful, but no one else can get this treatment. So Regeneron was an antibody, and the idea there was that we could generate enough antibody that we can throw that into your body, and we basically bind it to the virus that's intact, and it scoops it up, and then your body just destroys everything that has lots of antibodies attached to it. So the virus envelope is a possible target. What other things can we target to try and kill a virus? So you need to think about the virus life cycle. I know this is something that all of you are at least somewhat familiar with. It's not chemistry, it's biotechnology, really. So we can target virus replication. These are the nucleic acid handling proteins. So, viruses need to duplicate their genetic material. And so they've got RNA polymerases. If they're an RNA virus, if they're a DNA virus, they might have a reverse transcriptase to turn their RNA into DNA, insert it into the host's DNA, and then get the host DNA to spit out the RNA product. So you can target the reverse transcriptase. This is actually the basis of a lot of therapies for HIV. And it's because viruses polymerases are different than human polymerases, or even bacterial polymerases, eukaryotic polymerases. So they're very distinct, and that gives us a different target. You want to be very careful targeting polymerases. That's why this doesn't work for targeting funguses. Funguses scare the shit out of me because funguses are so much like us. We don't have a lot of ways to kill them without killing ourselves. But viruses, we can target their DNA replication machinery. We can target their protein replication machinery. So once you have the DNA then, or the RNA then needs to be used to generate new proteins. Now, generally that uses our host ribosomes, and there's nothing you can do, right? You do not want to inhibit the ribosome. You're dead if you inhibit the ribosome. But viruses aren't very clever, or maybe they're very clever. You don't know. So what they do is they tend to ask the ribosome to sequence one long piece of protein. And viruses make a protein that cuts proteins. So viruses make a protein that cuts them up into the functional proteins. So the initial protein that gets generated from the ribosome is really, really long. Then they generate this other little protein. It's the only other one they make. So for SARS-CoV-2, for example, it's the viral main protease. And it then slices up the big one into the individual proteins that the virus needs. So we can target that protein that cuts up the other proteins. We can target assembly, because the virus then needs all those little proteins to kind of come together to make a new viral capsid, get the DNA into it, and get ready to be shoved out. If you can screw up how those things can come together, if you can inhibit the formation of the complex, then you have a drug. There are a bunch of drugs against, some emerging drugs against Ebola that seem to be doing this, which is kind of cool, but it's a very difficult target. because we're trying to inhibit protein-protein interactions, and that is tough. These are very, very favorable interactions because viral capsids need to be very stable. And the other thing we do is we can inhibit virus release. So if we can stop cells from releasing the virus, so if we can keep, so the virus needs to, normally these viral capsids, what they do is they integrate and they bind to a membrane. Then the membrane vesicles get pushed out to the surface of the cell. They open up, they fuse with the cell membrane. The viruses are now on the surface. They then cut their bind to the membrane and they release themselves into solution. And if you can inhibit that cutting, then you can stop them from being released. So they're just stuck on the cell surface and they can't get off. And then we can inhibit viral entry. Because if once the viruses are free, the viruses can't get into the next cell, then they're stuck, they can't get in. This is a very common target, viral entry, because it seems like we understand it pretty well. So if we think about influenza, we often give it like we give it an HN number, all the influenza strains. So you have something like H1N1. Anyone know what the H and the N stand for? So the H is hemagglutinin. The N is neuraminidase. So these are both proteins on the viral shell. So the hemagglutinin is blood hema, glutenin binding, blood binding protein. And so it binds to cells, not just blood. It's more blood than that, but it was first found with blood. And depending on what subtype of that you have, it binds to slightly different receptors on cells. So which H value your influenza strain has tells you its specificity for specific cell types. The N value, neuraminidase, the A's gives you a clue that this is an enzyme. It's an A, so it's things that cut things, and it cuts neuraminidase, neuraminic acid. And neuraminic acid is a sugar. It's also known as sialic acid. So we haven't looked at sialic acid yet because it's a weird sugar and I don't really like it. It's a glucose derivative. It's an acid because, hey, look, there's an acid. Just look at this messed up molecule though. So it's obviously not a hexose. It's got way too many carbons on it. It's an extended form. And there are slightly different versions of it with slightly different stereochemistries and oxidation states on the side chain, whether this is acetate protected or not on the nitrogen. So they're all different names. Well, one I've drawn here is sialic acid. And what neuraminidase does is the virus uses the hemagglutinin to bind to the cell. Then it is coated with these sugars with sialic acids and uses sialic acids to kind of anchor itself. So, you know, the hemagglutinin forms a weak bond, then it sort of glues on with the sialic acid residues. And the reason those work is because our cells are covered in these lectin molecules. These are proteins that bind sugar. For recognition. So we talked about this at the beginning of the class. We're saying what are sugars good for, and one of the things is cell-cell signaling. and that is mediated by lectins. I think we're still at the very, very beginning of understanding how all these lectins work. I think our best understanding of lectins actually is in these kinds of mechanisms where we're looking at these kinds of drugs. So this is a sugar. It's a weird sugar. It's still a sugar. So it wouldn't surprise you to know that there's an R group there attached to other sugars. And what neuraminidase does is it cleaves this by using a glycosidase. And like all glycosidases, it goes through an intermediate that is pseudo carbocation. Not quite. We've seen that there's a lot of those are SN2 like, but they're all kind of still like a carbocation unit. They are actually SN2. And there's this chain over here. So I want to talk about this for a second. So if this is the intermediate as you're cleaning neuraminidase, then if you can block neuraminidase, if you can just mess it up, you can get a drug in there that's binding to neuraminidase, then we've got a shot at preventing viral entry. I'm going to redraw this as a hexagon kind of form of a sugar. I apologize. I should never do what I just did there. Drawing steric. There's no. How can I do this without ruining everything? I guess that's okay, but I don't like it. So if we can make a molecule that looks a bit like that, then maybe we can target that. So the problem is its transition state. But if I just replace that oxygen with a carbon, suddenly that's not a transition state anymore, right? It's got a very similar shape, but now it's perfectly stable. Replace that carboxylic acid over there with an ester. Why? Well, a couple reasons. One is it's greasy. And when you have greasy things, they tend to isolate into enzymes. Water is hydrophilic. Enzymes are always hydrophobic relative to water. So whenever you increase the greasiness of a molecule, you increase the amount of the time, you increase its ability to bind to enzymes. That's just a truism. We find that if we replace this oxygen with a nitrogen, we get a better binding strength of H bond. So there's an important hydrogen bond that oxygen is involved in. It helps make sure that you're cleaving neuraminidase and not something else, or neuraminic acid, not something else. So you need to make sure you have the right stereochemistry. So that OH is making a hydrogen bond with amino acid in the enzyme, and H2 makes a stronger hydrogen bond. So we're upping the ante on here, especially because it's probably going to be protonated under a physiological pH. So it's probably NH3+, and that's going to make a really strong hydrogen bond. We keep the NAC, we don't change anything. And we get to simplify the crap out of this thing. There's something wrong in my structure I have here. Just give me a quick second. Yeah, that makes sense. So we get the right stereochemistry here, but notice again what we've done is we've gone greasy. We have again this kind of big group here. There's a big pocket, and we're actually going to fill that pocket better because this is linear, so it can only go one way into an enzyme pocket, whereas if the pocket is very big, fills big pocket better. And again, what we've gone is gone greasy. So we've gone greasy in two places. We've actually decreased the greasiness, so we've increased the hydrogen bond on one place. We've kept the stereochemistry intact all the way around, right? There's something attached at every site. This is Tamiflu. So this drug is a transition state mimic of neuraminic acid that sits in a neuraminidase binding site. And when you sit back and you look at the molecule and you look at the transition state of neurominic acid, you can sort of see where that comes from and why this drug was designed. What I love about this was not developed through a particularly rational process. It was a high throughput screen of molecules, and Tamiflu, something related to Tamiflu was the thing that kind of jumped out at them as a good target, and then they developed it further. It's only in retrospect that we look and go, hey, look how similar these things are. Because they were just trying to find something that stopped the virus replicating, because it wasn't trying to block this particular mechanism. And this is one of the examples where we're looking at it going like, okay, maybe we need to think about how we design drugs, because this was designable. This was rationally designable. You can figure out that this is what you want to make by knowing something about sugar chemistry and something about enzymes. Not a lot. We haven't talked that much about it, but if I gave you a transition state mimic and I gave you some semblance of what the enzyme looked like, I expect you to be able to guess at something you think might be a good hit candidate for a drug, something that would actually work a little bit. Might not be great, might need more optimization, of course, but something to start with. So, cool. Transition state mimics, useful. Okay. Notice is also like a carbon sugar. We've removed the oxygen in the ring, so we talked about carbon sugars a little while back. That is a carbon sugar. We want to start talking about proteins and peptides. I'm not sure we did this exercise at the beginning of the course. I don't think so. But what's your working definition of a protein or a peptide? If we did it earlier, we're doing it again.
Working definitions, so how they function differently?
How do you define them? Everyone's got a different, like, I can look up the Merriam-Webster dictionary and find a definition of a protein. Actually, let me do that right now. I don't think it'll be a very useful definition of a protein.
Well, we actually use these two words quite interchangeably, don't we?
We do. So let's talk about how we can. So the definition of protein, according to the Merriam-Webster dictionary, is any of various naturally occurring extremely complex substance that consists of amino acid residues joined by peptide bonds containing the elements carbon, hydrogen, nitrogen, oxygen, usually sulfur, and occasionally other elements such as phosphorus or iron, and include many essential biological compounds such as enzyme, hormones, and antibodies. That is the definition of a protein. That is not a working definition. That is useless.
It doesn't make any sense to
It
me.
makes no sense at all. So that if we look up what is a protein, that is I just gave you the literally the fucking dictionary definition. It is useless. So we are going to take the dictionary and we're going to throw it out the window because it's useless. So what is a protein?
Protein is consists of amino acids. I think that's a definition we learned.
Yeah, I'll built from amino acids. I will take that as part of the definition. Are peptides also built from amino acids?
We use these words interchangeably for a reason, I guess.
I don't actually think they're interchangeable, and I actually do think there is and maybe we'll get to it. I do think there is a stark difference in definition. So I would say that something is either a protein or a peptide, not both a protein and a peptide. And I think I'm very comfortable defining where I define the line between the two. I think these are mutually exclusive categories. But proteins are often called polypeptides, whereas a peptide is obviously a monopeptide. So we'll start us off there. Okay, so proteins are built from amino acids. What more can we say about proteins? According to this definition, phenylalanine is a protein because it is built from amino acids. It's a single amino acid, but still built from them. Or if we're going to get, if you want to have plural, then diphenylalanine, so two phenylalanines stuck together, is a protein. And that's ridiculous. So what else goes into making a protein?
It's a 3D structure.
Yeah. I think you have a defined, if dynamic, 3D structure. So I think a protein has to have a specific 3D structure. And what I mean by specific, it's not frozen, it's constantly moving, but it's moving within a constrained conformational space. It can't just do anything. And some parts of the protein are going to be very, very, very ordered, and some parts are going to wave around randomly, but the whole thing doesn't wave around randomly. Okay, I think that's part of the definition. Building on that, is that 3D structure important for something?
Very important. It's usually determining for protein's activity and functions.
Perfect. So if the protein is misfolded, if that 3D structure is incorrect, the protein is generally not a functional protein. We would say it's a denatured protein. So you need the correct three-dimensional shape. Anyone else want to add anything else to this on the protein side? I think the only thing I would maybe consider adding is has a well-defined purpose. It can be an enzyme, it can be a receptor, it can be a structural component, but a protein exists to do something biologically. Okay. I think this is where we see the big difference with the peptide. So it also, proteins always have segments always have well-defined secondary structures. So you'll have alpha sheets, you'll have beta sheets, you'll have beta turns, you'll have polyprotein type 2 helices, alpha helices, hairpin turns, whatever. You'll have these handful, and it literally is a handful, it's under 10 defined secondary structures that will go together, be arranged in a unique three-dimensional pattern to give the protein its unique form. It's like, I always think of proteins kind of like Lego. There's only so, except there are a whole lot more Lego building blocks. There are only so many building blocks, but we can attach them together in nearly infinite ways to get infinite shapes. It's just unlike Lego, proteins are a lot more flexible. Lego building blocks are not very flexible once you've built something. Yeah. As my younger brother kept proving by breaking my Lego. I have never forgiven him. So, it's actually probably one of the reasons I became a chemist is because I'm doing like Lego with molecules instead. Unresolved childhood trauma. Anyways, peptides. What do we have? What part of this protein definition do you think we drop when we discuss peptides? If you're shy, you can put it into the chat. Come on. Someone other than Hazel. She's carried this lecture so far.
I actually have a question about the name peptide. So does this come from the peptide bond or the other way around?
I don't know. That is interesting. Let us take a look at what the dictionary says. This is where the dictionary might be useful. It might give us an etymology of the word. Peptide.
It's just that it doesn't really make sense to me because when there's a peptide bound, that means there's at least more than one peptide. Amino acids, I mean.
Yeah.
It just
So peptide comes from the word peptone, which is a noun in German, and that came from the Greek pepton, which is the neuter form of peptos, which is cooked or digested. So peptide comes from cooking in Greek. So where did this come from? Because it's got to do with the so that it was a German word for nitrogenous components of food, was pepton. Now, where does the word protein come from? Because wouldn't that make sense to be that? Okay, so it looks like protein comes from protos, which means first in Greek. And so protein was considered like the theoretical concept of food essential for life. The first requirement for life was protein, whereas peptide was the cooked substance. So it looks like these words have somehow made it into science completely individual of each other. So it looks like the peptide bond came from peptide, the substance, but we probably didn't know what peptides were when we started calling things peptide bonds. It was the thing that held peptides together, but we didn't know what peptides were. And then when we figured out what that was, we probably went, oh, it's the amide bond within a protein is a peptide bond. So I think these one is kind of this unknown element that is in food, and one is like cooked nitrogen. I think this requires a lot more than some quick Googling on etymology websites. That's a really interesting question, Hazel, and I think I'm going to look into it because it sounds like the kind of rabbit hole I'm going to waste time on.
Yeah.
Okay. So it looks like peptide bond comes from peptide. But a peptide bond is also in proteins, which is confusing.
Yeah, I asked that question because I thought it would help me to think, but I guess no.
No. Originally, protein was actually used for all sorts of organic biomolecules, like nucleic acids were proteins and so were sugars. Everything was a protein. Interesting. Okay, anyways. That would have been confusing. Okay. So I draw the line at peptides do not have a well-defined three-dimensional structure that's required for function. At least natural peptides don't. I think we are getting a little. There's a little bit of a wet area where I have these ginormous peptides that are kind of like subsets of proteins. They're not independently fully functioning things, but they're kind of like truncated versions of the protein. I think that's a little bit of a gray area. But most peptides don't have well-defined three-dimensional structures. They might have a very well-defined secondary structure. They might form an alpha helix, they might form a beta sheet, that happens, but they don't form a well-defined complex three-dimensional structure that gives independent function. And so they can't operate independently. They can't be a receptor. There are no peptide receptors. A peptide receptor is a protein that binds peptides. It isn't a peptide that acts as a receptor. There are no peptide enzymes. There are no peptides that act as enzymes. Because to be an enzyme, you need a well-defined three-dimensional structure. Peptides lack a well-defined three-dimensional structure, so peptides are not enzymes. Some peptides are acting as structural components, but that's when they massively aggregate into really large complexes. And you have this it's not actually the peptide that's doing it, it's this big construct. And so we see that with Alzheimer's disease is basically a result of amyloid peptides, not the amyloid proteins, but the amyloid peptides degraded from amyloid protein that start to aggregate together and bind together. Amyloid protein behaves just fine. It's when it gets cut up into shorter sequences that lack well-defined three-dimensional structures, and then they all kind of band together and form these plaques, and you're screwed. So the operational definition between these things is proteins have well-defined three-dimensional structures that define their form and function. Peptides lack well-defined three-dimensional structures, and so their function and form is defined by their interaction with proteins. So roughly speaking, I'm not aware of any protein that is under 50 amino acids. There are a couple of really small ones which are right at that borderline of like 52, 53 amino acids. The vast majority are well over 100. The vast majority are, like, most are over 200 or 300 amino acids. A few thousand is not unusual. Whereas peptides, I would say an operational definition is between 5 and 50. Under 5, we're normally talking about things we call oligopeptides, just to confuse the issue. Because to me, an oligosaccharide is many saccharides, like 4, 5, 6 saccharides, oligo, several. Whereas an oligopeptide, you think, okay, it's several peptides. No, it's a short version of a peptide with several amino acids. We don't call it an oligoamino acid, which is what we should call it, because nomenclature is stupid. So peptides are between about 5 and 50. Under 5 is an oligopeptide. Normally, no function. I'm not aware of a biological function for any peptide that is under 5 amino acids. The smallest ones that are normally functional are up around 10 amino acids, and those are functional in the immune system. There are a few really, really short hormones at about 7 amino acids, but I can't think of anything 5 and under that has any function. It doesn't mean they're useless. It just means they have no function. We find uses for them, like, alanine, phenylalanine is a sweetener. That's aspartame. We find it really, really sweet. That's really, really useful for us, at least for the chemical industry. So just because it's really, really short doesn't mean it's useless, but it means it doesn't have a biological function. So my Pepsi is sweetened with aspartame, so I do think short peptides are really, really useful. Because I down this stuff like it's Pepsi. Pepsi, of course, does share a it is related to peptide, actually. That's where the name actually comes from. Just because I think peptides were cool at the time. It's got nothing to do with the artificial sweetener that came in the 60s. So let's just take a think about, these are all made from amino acids. What's the definition of an amino acid?
A molecule that has an amino and acid.
Yeah, that is literally it. That's it. That's all. That's what an amino acid is. So this is alanine, as you guys know. We would call this an alpha amino acid. And that's because that is the alpha carbon, and the amino group is attached to the alpha carbon. It is alpha because it is the carbon adjacent to the acid. We can have beta amino acids. This is beta serine. It's an amino acid, but it is not an alpha amino acid. And so when we talk biology, we're almost always talking about alpha amino acids, but amino acids are a broader category. Then you can have like weird ass amino acids. That's an amino acid. I have one too many carbons. 1, 2, 3, 4, 5, 6. This is a building block for nylon. It's an amino acid. It's just not a biological amino acid. So just be careful about the terminology. I'm going to use the term canonical amino acids. To refer to the 20 eukaryotic biological amino acids. There are a few edge case ones, like ornithine, which isn't a canonical amino acid, doesn't make up our proteins. It's not a proteogenic amino acid. These are also the proteinogenic amino acids. So these are the amino acids that make up our proteins. Ornithine doesn't make up our proteins. It's a version of lysine where we remove one of the CH2s in the side chain. We have to be careful with this because there are some modifications to the amino acids that occur in our proteins, like if you want to look it up, asparagine can become citrulline. Citrulline is definitely not a proteogenic amino acid. It's not one of the ones we use. You've got a lot of citrulline attached to amino acids in your body. So it gets a little gray, but I will refer to the 20 guys as the proteogenic ones, are the canonical amino acids. Everything else is a non-canonical amino acid. All the canonical amino acids, of course, are alpha amino acids. Okay. So how do you make a protein? Let's say I say here, make some P53. P53, guardian of the genome, important protein, does a lot of things. And it's about 387 amino acids. I'm actually going to look up exactly how long it is. I think I'm underestimating the size by quite a bit. I think I'm just thinking about the core region. It's very, very big. It's got lots of amino acids. So let's say you're in the lab and somebody, working at a company, working on a project, and they're like, okay, we need some P53 for this project. How are you going to get them P53? How would you make this?
Definitely not the chemistry way.
Definitely not the chemistry way, exactly. So how do you make protein if you're not making the chemistry way?
Biology way.
Yeah, okay. So generally, you know, transfection or plasmid into competent cell line. So you're going to take a plasmid coding DNA into a competent cell line. So your competent cell line might be mammalian, might be insect, it might be bacteria, depends on what you need for the protein to do. And you basically hijack the machinery of the cell line to generate the protein, right? It's like the fundamental tool of biotechnology and chemical biology. Okay, this works if all residues are canonical, right? Because bugs make bug proteins. And we can modify it. We can genetically engineer the plasmid to express different amino acids at different sites. It's not really hard. We do it in I think we do it actually in our biotechnology undergraduate labs. I think we actually engineer proteins in that lab. I did in a biotechnology undergraduate lab, so it's trivial. But this gets really, really tough if some of the residues are not canonical and you want to introduce some unnatural amino acid residues. 25 years ago, there was no way to do it. It was impossible. It's now possible. So what you can do is we can use engineered stop codons. So we have these engineered tRNAs that recognize stop codons and carry unnatural amino acids. So stop codons in a gene are a set of 3 nucleic acids that do not code for any protein, and the reason, or amino acid, and the reason they do not code for any amino acid is because there is no transfer RNA molecule that can recognize them. So what's been done in the last 25 years is people have actually engineered new transfer RNA molecules that recognize the stop codons. And so instead of being a stop codon where translation stops, you stop making protein, what we get now is it finds a tRNA, which then loads another amino acid on. And so we can customize, we can actually, engineer biology, synthetic biology, to incorporate unnatural amino acids into this. But it's still very limited. Still really hard to do. And there's only a handful of artificial tRNAs for a very handful of unnatural residues. So an important part about all this with the protein is you can add a purification tag. It does not say purification, does it? And so like a his tag, like a histidine tag is very common. So it's a sequence of 5 to 9 histidines in a row at the very end of the peptide, or the protein, sorry, and that binds really well to nickel. So we can basically pull that protein separate from all the other proteins present. That's really important because, the cell is making all these proteins. It's not just making your protein, it's making all of its other proteins. You need to get your protein separate from all the other proteins, so you need to add some sort of purification tag, or you need to find some way to purify your protein. Now, if you're making a small peptide like 20 amino acids long or 10 amino acids long, attaching 5 to 9 histidines to that is probably going to affect what you're interested for that peptide to do, because your peptide is really short. So if half of its size is a purification tag, good fucking luck. And again, because it's so short, it's going to be really hard to separate from all the other crap in the cell. So biology sucks. This. It's really hard to make and isolate small peptides from biological methods. So we use chemical methods. Okay, so I think, really early, what we're making is we're trying to make peptide bonds, right? And a peptide bond, real simple, where R is just stuff, whatever. Right? Plus water. It's a condensation reaction. We bring an amine and we bring a carboxylic acid together and we make a peptide. We're going to spend a bit of time talking about the different ways that we can do this. Just like glycosylation was all about what was going on with the anomeric center, this part of the course is all about what's going on at the peptide bond. But I want to just think about what the challenge is. So let's say I want to make alanine, glycine. If you don't have a table of the 20 amino acids and the 3-letter codes and 1-letter codes, I would recommend you go online and find one. I'm never going to ask you what is alanine, because you can look it up. I'm going to assume you know what alanine is, or I'm going to assume that you can look up what alanine is. And we're going to talk a little bit, one of the assignment questions coming up on the assignments for the final discusses, hey, look into the protecting groups for these residues because they're important, and draw them all out and think about how you deprotect the standard ones and why they're used. So I'm just going to use the 1-letter codes or 3-letter codes as are more appropriate for a given application. But just as a reminder on how to read this, we always read from the N-terminus to C-terminus. So saying AG like this can be written that way. The way you will also see this occasionally is people will explicitly say, hey, look, it's the N-terminus and the C-terminus. And occasionally I'm actually going to write them backwards. And I promise there's a rationale for that. And if I write it backwards, I must include the fact that my N-terminus is the NH2 side and C-terminus is the carboxylic acid side. But if we just write it using this code, we are always writing from N to C. That is the convention. We read left to right. We write peptides and protein sequences left to right from N to C. Just is. So if I'm thinking about what that structure is, why I write AG, because it's a whole lot shorter than writing this. It's that. So I want to make this my peptide bond. I want to do a disconnection there. This came from alanine and glycine. Okay, so that retrosynthesis arrow is relatively simple. But let's say I have magic reagent. For now, let's just put magic. So if I mix these two together, magic reagent, that makes peptide bonds. Am I going to get AG? So I mix a 1-to-1 mixture of these two together. This is where I'm ending today, so we can, stare at each other, or somebody can think about this. I can stare at the black boxes, and you can stare at me. I got coffee to drink though, so I'm happy.
I guess this really means no one knows.
Yeah, I don't think anyone's answering this. This is not an inspiring course to teach this cycle. Most years it's a really, really huge discussion. Everyone's interrupting me constantly. You guys are a really quiet audience. It's distressing. I love this class so much. Okay. The answer is no, you don't. We'll start next class with a poll where everyone needs to vote on what they think the product of this is. You're not getting AG. Well, you are, but you're not only getting AG. Okay, let's leave it there. It's 9:50. Have a good rest of your Wednesday. I have no idea what day of the week